All right, so there are two types of transport. There's active transport and passive transport. And today we are going to talk specifically about passive transport. Wait a second. Passive transport refers to any type of movement of molecule that does not require energy. Okay, so in order for there to be movement of molecules that doesn't require energy, they have to move down a concentration gradient, which means they have to move from an area of high concentration into an area of low concentration. So we'll say that molecules must move from high concentration to low concentration. Okay, so this term, high concentration to low concentration, can be summed up by saying the words concentration gradient, or saying moving down a concentration gradient. Okay, and so what that means is a concentration gradient you can think of like a ramp. Okay, so if you had a couple solutions, let's say you had a 5 molar solution, you had a 4 molar solution, a 3 molar solution, a 2 molar solution, and a 1 molar solution, right? You could just stack up 1, 2, 3, 4, and 5. 1, 2, 3, 4. 1, 2, 3. 1, 2, and 1. And you can see how this goes through a nice little ramp here, right? Okay, so if we talked about this little dot at the top here, okay, this little dot at the top here could roll down this ramp like this, right? Would that require any outside energy for it to roll down that ramp? No. Okay, what if we wanted to take this dot here and move it up? Would that require energy? Absolutely it would, right? And that's what we're talking about when we say a concentration gradient. It doesn't require energy to move down, right? If we go this way, this is passive. Just like moving down a ramp does not require energy, but moving up a ramp does require energy. Now there's 3 types of passive transport that we're going to look at. The first one is called simple diffusion. Simple diffusion means that molecules are going to go from an area of high concentration to low concentration, and they're going to do that because they are permeable to whatever substrate they're in. Okay, so we can say molecules move from high concentration low concentration through a permeable substrate. What does that mean? Well, if we're talking about the room, air is permeable to a lot of different odors, right? So if I sprayed some perfume over here, it wouldn't take very long for it to sort of permeate the room, right? But it would never be as concentrated when it gets back to that back corner as it was when I sprayed it up here, because there was a high concentration here, and then it goes through diffusion and it diffuses until it's at equilibrium. The point where it's at equilibrium is where it's an equal concentration throughout the entire room, right? You guys have all heard this a million times? All right. When we talk about simple diffusion for the plasma membrane, what are we talking about? What type of molecule? Small nonpolar only, yeah. Small nonpolar molecules are the ones that are easily going to be able to go through simple diffusion. Everything else is going to need some help. So if it needs help, it's going to go through something that's called facilitated diffusion. Facilitated diffusion means that molecules are going to go from an area of high concentration to low concentration, but they are going to do so with the help of some other type of molecule. Looks like through a non-permeable substrate. With the help of another molecule. Now, what other molecules might help here? What are the molecules that we just learned about? Yeah, channel proteins, carrier proteins. Permeases. Right? And so basically anything can move through the phospholipid bilayer as long as it's going from high concentration to low concentration and it has a carrier protein or a channel protein that's associated with it. Right? Okay. Last thing, probably the most misunderstood of all the types of passive transport is osmosis. You ask people if they know what osmosis is, they're like, yeah, I know what osmosis is. And you're like, okay, what is it? Osmosis Jones? You guys have seen that movie?
Yes.
It's a good movie. All right. So, maybe it's because, so when I was in middle school, there was this really popular poster. I think like maybe it got sent out to every teacher ever in my entire school district because it was like in every single classroom. And it had Garfield on it, and Garfield had a stack of books on his head, right? And underneath it said, "I'm learning through osmosis," right? It's hilarious, right? But it's so wrong. It's terrible, okay? Because you can't learn through osmosis because osmosis is the transport of water, right? So we're gonna say the movement of water from an area of low solute concentration to an area of high solute concentration. So let's entertain this poster for a second, okay? So it says he's learning through osmosis, so we can consider learning like he's talking about the solute as like knowledge, right? That's what he's saying is moving into his head through osmosis, which is wrong, first of all. But the second problem is that Garfield would literally die if that happened. And the reason for that is that he's got books, and you can pretty much assume that the books are going to have more knowledge in them than Garfield's head, right? And so Garfield places the books on his head that have more knowledge, and that means water is going to be pulled from Garfield's brain into the books, and Garfield dies. It's a morbid poster when you really think about it. And so this is different than everything else is talking about. So the different types of diffusion, we always wrote moving from an area of high concentration to low concentration, but here we wrote we were moving from an area of high solute of low solute concentration into an area of high solute concentration. So what? That seems backwards, right? It does. And the reason for that is that we're talking about the movement of water, and water is not the solute. Water is the solvent, right? And so we're still saying that water is moving from an area of high water concentration into an area of low water concentration. But that's not how we measure solutions. We measure solutions based on the other thing, based on the solute, not the solvent. And so we say, okay, if something is really high solute concentration, that means its water concentration is low. If something is really low solute concentration, that means its water content is really high. Right? So the water is still going from high water concentration to low water concentration, but that's not how you describe the solutions. Does that make sense? All right, let's do some drawings to try to clear that up. So here's a beaker. Here's a beaker. These are not the same size. All right, so 3 beakers. In these beakers, they are going to be separated in the middle. There's a membrane in there. Let's say that membrane is similar in its properties to a phospholipid bilayer. Okay? I'm going to put some water in. This. Over here, I'm going to put less water in. I'm going to put this much water in. I'm going to put some different things in here. I'm going to put O2 in this one. O2 over here. I'm going to put H plus ions in here. And over here I'm going to put Na plus ions. Okay. So they're all the same. There's 5 on the left side and 1 on the right side. So we're going to say this is like 0.5 molar O2. This one is 0.1 molar O2. This one is 0.5 molar H plus. And this one is 0.1 molar H plus. This one's, if we ignore the fact that there's less water, which we're going to, 0.5 molar Na plus and 0.1 molar Na plus. Okay, so just like this, what happens to these beakers? Do any of them see any change? Which one would change? If we call them beaker 1, 2, and 3, which beaker changes? What do you think?
2.
Why?
Because it'll diffuse from high low.
Okay. Nope. What do you think?
1.
Why 1?
Because it's not 2.
Because it's not 2. All right. Why 1? Because it's nonpolar, right? So, in beaker number 1, if we're going through a phospholipid bilayer, the only things that can go through the phospholipid bilayer are small and nonpolar, right? And so that means that this diffuses into here like that. Okay? This is called simple diffusion. And you'll end up with a beaker that looks like this. Where you now have an equal amount of O2 on both sides, which was 0.3 molar then. So they've equaled out, so now they're 0.3 molar each. What's the movement look like now? Do they move at all? They do. But they move equally, yes. So that's the big misunderstanding, misconception that people have is they say, oh, once this is in equilibrium, the molecules stop moving. No, they just move at equal rates. So whenever a O2 diffuses across the membrane this way, an O2 will diffuse across the membrane that way equally. Okay, so nothing happens in these other two. Let's say I throw some ATP synthase in here. This becomes ATP synthase. Now what happens? Yeah. They'll equal out to the other side because now they're going to go through the ATP synthase. Before they couldn't go through because they were polar, but now they can go through because there is something facilitating it. Therefore, we would call this facilitated diffusion. And they will equal out so that there are plus ions there. Totally unrelated. How else could we describe the concentration of H plus ions? What other term can be used to describe the concentration of H plus ions? What do you think?
High potential energy?
Nope. Chemistry term? pH. pH describes the concentration of H plus ions. So if you took the negative log of 0.3, its pH would be 0.522. You guys remember that in chemistry? All right. All right, so that's facilitated diffusion. Next, what happens in this one? Nothing happens. But what about if we put in some aquaporins? Now that aquaporins are in there, now what happens? What? Nope, not facilitated diffusion. Osmosis. So what happens? What happens in osmosis?
The water from the Na plus side that only has one of it will move over so that the concentration is like at equilibrium.
Yep. So our concentrations are going to have to equal out. And the only way to equal out the concentration, since the Na pluses can't move, is to have there be significantly more water on this side than there is on this side. So we still have the same amount of Na pluses. But the water has moved. And this process is called osmosis. And then we would say there's an equal concentration of 0.3 molar and 0.3 molar or something like that. Make sense so far? No, I mean, technically this doesn't work because if the aquaporins were like here, where I guess I drew them at a different spot. But I drew them like here, right? But this wouldn't really work because this side of it technically has no water at all, right? Because it's out of the water, so it would start pouring back out of there, but that's okay. We'll pretend like it doesn't. All right, so 3 terms now. We're going to use 3 terms to describe things like this. One of the terms is hypotonic. One of the terms is hypertonic. And one of the terms is isotonic. Okay? Another way for saying concentration is to say the tonicity of a solution, right? And so that's where this tonic thing comes from, is tonicity. It's talking about its concentration. And then we just need to be reminded of the prefixes here. So there's hypo, hyper, and iso. Hypo means below, hyper means above, and iso means the same, right? We talked about isomers, right? We talked about isotopes, right? Again, this is isotonic, which means the same. Hypo means below, so that means that something that is hypotonic has a lower solute concentration. Something that is hyper has a higher solute concentration. And something that's iso has the same solute concentration. Now, I want you to notice these words right here. Lower, higher, and the same. Those are all comparative words, right? I can't show you a single beaker and say, is this isotonic, hypertonic, or hypotonic? Because you can't, that's not a thing. It has to compare it to something, right? But we can say up here, we're talking about the 2 sides. We can say, this, if this is side number 1 and this is side number 2, we would say that side 1 is hypertonic to side 2, right? We could say side 2 is hypotonic to side 1, right? So this side would be hypo, this side would be hyper. Then we get down to the bottom. These are isotonic, right? Because they both have the same solute concentration. Does this make sense? Anybody have questions on it? All right, that's it. Yeah.